Biotechnology deals with the application of biology and biological concepts to science and engineering for the welfare of mankind. It is the crossroad of biological sciences with other major disciplines of science, chemistry to mechanical engineering. The history of biotechnology as old as the history of man. When the first human beings realized that they could plant their own crops and breed their own animals, they learned to use biology. But the term biotechnology was introduced by the end of 20th century. Nowadays, the field of biotechnology becomes a vast field, but this chapter touches a brief part of biotechnology called genetic engineering, which deals with manipulation or alteration in genetic material of an organism. Cloning of genes. Gene cloning is the act of making copies or clones of a single gene. Once a gene is identified and cloned, biomedical and industrial research. There are two possible ways of cloning of genes: recombinant DNA technology, polymerase chain reaction. Recombinant DNA technology. Recombinant DNA technology is a series of procedures that are used to join DNA segments from different sources. This is an in vivo and living cells method which is used when gene cloning is required at industrial scale. The main advantage of this method product of gene beside copies of gene. It involves the selection and isolation of desired gene of interest, inserting it as at a suitable vector and the transformation of a suitable host by the recombinant DNA components. Tool of recombinant DNA technology. The cloning of gene recombinant DNA technology requires gene of interest, molecular scissors, molecular carrier or vectors, molecular glue, and expression system. Gene of interest. The gene of interest is the gene which is to be cloned. It can be obtained by one of the three possible processes of synthesizing a gene in, in glassware without template DNA samples with the help of DNA synthesizer machine. Gene of interest can also be obtained by synthesizing it from its messenger RNA of gene from messenger RNA is carried out by reverse transcriptase enzymes, which are naturally found in retroviruses. DNA formed by this process is called complementary DNA. In most of the cases, the gene of interest is directly cleaved from chromosomal DNA by using particular DNA scissors called restriction endonucleases. Molecular scissors, restriction endonucleases. Restriction endonucleases are enzymes that leave phosphodiesterase sequence in 1970. Instruction enzymes are capable of destroying DNA bacteriophage by convention. Instruction enzymes are named after their host origin. For example, EcoR1 was isolated from Escherichia coli or Escherichia coli. Hind2 and Hind3 from Haemophilus influenzae and Zol from Xanthomonas hospice. Naturally, restriction enzymes are found in bacteria where they appear to serve as host defender make staggered cut or blunt cut. Staggered cut is one in which the resulting duplex fragments show single-stranded projected ends called sticky ends, while in blunt cut the resulting duplex fragments do not show such sticky ends. The recognition site is boxed in yellow and the cut site indicated by red triangles. Molecular carriers are vectors. Vectors are another major component required to make a recombinant DNA or DNA molecule for gene cloning. Vectors act as a vehicle for carrying foreign DNA into a host cell for multiplication usually. Small circular DNA molecules of bacterial origin are used as cloning vectors. A DNA molecule should possess the following essential characteristics to act as a cloning vector: origin of replication site, antibiotics resistant genes, destruction sites of different enzymes. Example of vectors are plasmids, lambda phage DNA, plasmid, combination of plasmid and phage DNA, yeast, artificial chromosomes, etc. Molecular glue, DNA ligase. This is and a foreign DNA that are annealed by the sticky ends. Expression system. A suitable organism that can act as host for the recombinant DNA vector to express multiplication is called expression system. Therefore, the selection of suitable expression system always depend upon the type of vector which is being used while making recombinant DNA. The most important character of an ideal expression system is its short generation time and simplicity of its genetic system. So bacterial cell can act as an ideal expression on system. Assertive cloning of the desired gene through recombinant DNA technology involves the formation of recombinant DNA gene of interest, vector DNA, transformation of a suitable expression system by recombinant DNA, and identification of transformed clones. Formation of recombinant DNA. The first step in the construction of recombinant DNA is the isolation and purification of vectors and genes of interest. First, digest the vector DNA, for example, plasmid, with same destruction enzyme by gene of interest as cleaved so that compatible sticky ends can be produced. Next, both vector and gene of interest are incubated together in the presence of DNA ligase, which connects them by forming phosphodiester linkage. This results in the formation of recombinant DNA molecule of vector and a gene of transformation of expression system. Here, transformation refers to the insertion of recombinant DNA into the expression system, which can be performed by putting the expression system bacterial cells that already contain no plasmids and recombinant plasmid into the same medium. Bacterial cells take up recombinant plasmid, especially if they are treated with calcium chloride, which make them more permeable. Thereafter, as the cell reproduces, a bacterial clone forms, and each new cell contains at least one plasmid. Therefore, each of the bacterial cells contains the gene of interest, which will express itself and make its product. Identification of transformed clones. The transformed clone can be identified by adding a particular antibiotic for which resistance gene is found in plasmid into the medium. As a transformed clone has got resistance against resistant gene is found in plasmid into the medium. If the transformed clone got resistance against antibiotic, it remains alive and continues to grow. Because the gene of interest is inserted inside the other antibiotic resistant gene, where all the untransformed clones are killed by antibiotic. From this transformed bacterial clone, the cloned gene can be isolated for further or its protein product can be separated. Polymerase chain reaction (PCR). Polymerase chain reaction as a revolutionary technique in molecular biology to amplify cloning a single or few copies of a piece of DNA to generate thousands of millions of copies. A single or few copies of a piece of DNA to generate thousands of millions of copies. It was originally invented by Carey Mullis in 1983. Later on, he was awarded the Nobel Prize in Chemistry in 1993. PCR is a process that amplifies or copies a piece of DNA repeatedly until there is an amount which is great enough to observe usually. It is based upon in vitro replication processes which is carried out by DNA polymerase enzymes. In this technique, DNA polymerase is compelled to polymerize a given piece of DNA again and again so that multiple copies are produced. Thus, the technique is known as polymerase chain reaction. Components of PCR techniques. The following are the components required for carrying out a PCR reaction: template DNA, deoxyribonucleoside, triphosphate, DNTPs, primers, take polymerase. Template DNA or target DNA, it is the piece of DNA to be cloned or amplified. It may be a useful gene found in the genomic DNA or the piece of DNA of infecting organisms. Deoxyribonucleoside, triphosphatase, triphosphate DNTPs. These are free nucleotides that act as raw material for synthesis of new DNA fragments. There are four different types of DNTPs: DATP, DGTP, DCTP, DTTP required in this process. Primers. DNA polymerase is suitable to initiate polymerization unless primers are attached. Two sets of primers, forward primer and a backward primer, are used in this technique that select three ends of target DNA fragments by annealing with its complementary sequences. Take polymerase. The take DNA polymerase as a temperature tolerant enzyme isolated from Thermus aquaticus, a bacterium found in hot springs. It catalyzes the synthesis of DNA. This enzyme is stable and active at near boiling temperatures. Therefore, it is well suited for carrying out PCR reactions. These are basic components required for the assembly of a PCR reaction. They form a mixture called PCR mixture or reaction mixture. The PCR mixture is placed in an instrument called thermal cycler or PCR machine. Thermal cycler regulates the temperature during various steps of PCR reaction according to the need. Mechanism of PCR reaction. A PCR amplification cycle consists of three basic steps. They are denaturation, primer annealing, and extension or polymerization. As time duration, temperatures, and sequence of the steps have to be programmed in thermal cycler. Denaturation. In this denaturation step, the template is heated to 94 degrees centigrade for 1 minute or up to 5 minutes. At this high temperature, the DNA undergoes complete denaturation and the double-stranded DNA, dsDNA, becomes single-stranded ssDNA. Each single-strand DNA can act as the template for the in vitro DNA synthesis. Primer annealing. The next step is the primer annealing, and this step, the two primer, the forward primers and the backward primers anneal to hybridize to the single-stranded template DNA at its complementary regions. Annealing is usually carried out at a lower temperature, depending on the length and sequence of the primers. In standard cases, it is 54 degrees centigrade, and approximate time required for this step is 2 minutes. Extension or polymerization. The final step is in each cycle is the primary extension or polymerization, in which the tech DNA polymerase synthesizes new DNA strand to the three end of primer using DNTP. The optimum temperature for carrying out the primer. Extension reaction or polymerization of DNTP is standardized at 72 degrees centigrade. This step takes just 1 minute to be completed. Mechanism of PCR reaction: target sequence, primer, new DNA, new DNA, single strand DNAs. At the end of first cycle, one target DNA molecule is converted into two molecules. The second cycle immediately starts with the denaturation by heating at 94 degrees centigrade so that all the newly synthesized DNA are also denatured to single strand which again act as templates. It will again be followed by primer annealing and extension and thus the cycle of denaturation, primer annealing and extension continues resulting in the amplification of the selected DNA sequence at an exponential rate. The number of existing DNA molecules become doubled after each cycle. Application of PCR. PCR has application in almost all areas of molecular biology, genetics, and in clinical areas. PCR is an efficient diagnostic technique used for the detection of specific infectious agents, for example, HBV, HCV, HIV. It is also used for the detection of microorganisms in food samples, water, and the environment with the help of species specific primers. PCR can also be used for genome analysis and for generating markers for the construction of genetic and physical makes of organisms. There as the PCR-based cDNA synthesis known as RT-PCR, reverse transcriptase PCR, which can be directly carried out with purified messenger RNA. Reaction for DNA sequencing are also simplified by introducing the PCR method. PCR has also shown its impact in criminology. The DNA of the suspects and the DNA sample record from the crime scene can be analyzed by PCR techniques with the help of a set of identical random primers of specific primers. DNA fingerprinting is also made simple by PCR as described above. The gene mutations responsible for certain genetic diseases and cancer can be detected using PCR tools. Early detection of genetic disease as even possible in embryonic conditions or even in sex cells, sperm, and eggs. Genomic library. The genomic library is a collection of bacterial or bacteriophage clones, each containing at least one copy of every DNA sequence and a genome of an organism. In a single library, the entire genome of an organism is respected the set of DNA finger fragment inserted into a vector molecules. Construction of genomic library. To construct a genomic library, the genomic DNA of the organism is extracted and is cut into fragments of suitable sizes by any of the following three methods. The genomic DNA is digested completely by restriction enzymes that convert it into fragments of suitable sizes. The restriction enzymes cut at all relevant restriction sites and produces a large number of short fragments with sticky ends. The disadvantage of this is that genes containing restriction sites within the reading frame may be cut into two or more fragments and may be cloned separately. The genomic DNA can be fragmented unenzymatically by means of mechanical shearing, such as sonication, which produces longer DNA fragments. The disadvantage in this case is that end of fragments produced are not uniform and need enzymatic modification for insertion into a cloning vector. The third method for fragmenting the genomic DNA is by partial enzymatic digestion with restriction enzyme. Locating gene of interest from DNA libraries. A DNA probe is small, fluorescently or radioactively labeled DNA molecule that is used to locate similar or complementary sequences among a long stretch of DNA molecule or bacterial colonies such as genomic or cDNA libraries are in a genome. DNA sequencing. To understand the complexity of gene structure, its expression, its regulation, protein interactions, and molecular mechanism of genetic diseases, the detailed and exact sequences of the bases in DNA is very essential. The main principle of DNA sequencing method is to generate piece of DNA of different sizes, all starting from the same point and ending at different points. Separation of these different sizes pieces of DNA by gel electrophoresis, reading up sequence from the gel. For generation of different size DNA fragments, two different sequencing methods were developed during the late 1970th century. They are Sanger method and Maxim Gilbert methods. Singer's method. This method is widely used and similar to the natural process of DNA replication. It was developed by Frederick Sanger along with Andrew Coulson in 1977. They were awarded Nobel Prize in 1980 on this achievement. Sanger's method now became the standard because of its practically procedure. Before the DNA can be sequenced, it has to be denatured into single strands using heat because only one strand that acts as template is required in this procedure. Now the template strand is checked with a known sequence at 3 int so that a complementary primer can be bind on the known sequence. Once the primer is attached to the DNA, the solution is divided into four tubes labeled G, A, T, and C. Then reagents are added to these samples as follows: G tube, all four DNTPs, GTP, and DNA polymerase. A tube, all four DNTPs, DDATP, and DNA polymerase. T tube, all four DNTPs, DDTTP, and DNA polymerase. C tube, all four DNTPs, DDCTP, and DNA polymerase. As shown above, all of the tubes contain different DDNTPs present, and each at about 100 the concentration of the normal precursors. Now, all these test tubes are placed in PCR machine so that sequencing reaction can start. At the DNA, a synthesized nucleotides are added on the growing chain by the DNA polymerase. However, on occasion, a dideoxynucleotide is incorporated into the chain in place of a normal nucleotide, which results in a chain terminating event. For example, if we looked at only the G tube, we might find a mixture of the following products. The key to this method is that all the reactions start from the same nucleotide and end with a specific base, thus in a solution where the same chain of DNA is being synthesized over and over again. The new chain will terminate at all positions where the nucleotide has the potential to be added because of the integration of the deoxynucleotides. In this way, bands of all different lengths are produced. Once these reactions are completed, the DNA is once again denatured in preparation for electrophoresis. The contents of each of four tubes are run in separate lanes on a polyacrylamide gel in order to separate the different sized bands from one another. After contents have been run across the gel is then exposed to either ultraviolet light or X-rays depending on the method used for labeling the DNA. Gel electrophoresis. Gel electrophoresis is a technique used in molecular biology to separate charge-bearing polymers, proteins, RNA, or DNA under the influence of electric field. DNA electrophoresis is used to separate DNA fragments primarily by size. The type of gel most commonly used for DNA electrophoresis are agarose for relatively large DNA molecules and polyacrylamide for high resolution of short DNA fragments. The freshly prepared gel is in liquid form, but when it is poured in a gel cast to form a thin slab of gel and is allowed to be cooled, it turns into solid state. At one end of the gel slab, some partial holes are made, which are called wells. They are partial in sense that they are not opened from other side of the slab. Later on, various samples of mixture of different size DNA fragments are poured into these wells. The gel slab is suspended in a buffer solution, which is used to establish electric field. The buffer used for the separation are trace borate EDTA or trace acetate EDTA. To establish an electric field, a positive electrode at one end and a negative electrode at another end of a solution are inserted. As the electric current runs between the electrodes, the DNA fragment of different length present in the well begin to move from negative pole to positive pole because DNA molecules have negative charges due to the phosphate groups. The DNA fragments migrate relatively to its size because the distance a DNA fragment travels is inversely proportional to its length, so the smaller fragments move faster through the gel matrix than larger fragments. Although the movement of the fragment also depends upon charges, number of strands, single or double, shape of the molecule, linear or circular, and concentration of the gel force size. When the movement of fragment is stopped, the DNA molecules are appeared as band at different position of gel, but still bands cannot be waved until they are labeled with fluorescent dyes or radioactive probes. Following the separation, the gels can be stranded by DNA binding fluorescent dyes that bind to the DNA molecule and are typically waved under ultraviolet illumination. DNA bands can also be transferred from gel to the nitrocellulose membrane for autoradiography, X-ray imaging. In the image of gel pattern, some bands are appear thick and some are thin. Thick bands represent the high concentration of same-size fragments, while thin bands show low concentration. If a particular size fragment is to be used for further analysis, the piece of gel containing that band can be cut and the DNA can be purified again. Automated DNA sequencing. Automated sequencing machines have greatly improved the quality as well as the speed of sequencing process. The basic principle sequencing is quite same in manual and automatic DNA sequencing except few differences. There is no need for radio labeling and autoradiography. The use of fluorescently labeled DDNTPs, dideoxynucleotide triphosphate, has made the reading very easy, convenient, and automatic with the help of ultraviolet laser detectors. Thus, it has greatly improved the speed of sequencing. Each of the four types of DDNTPs can be labeled with a specific dye so that a specific color can be attributed to the presence of a particular nucleotide or base. After electrophoresis, we don't even have to read the sequence from the gel. The computer does that for us. After electrophoresis, the colored band can be monitored using ultraviolet laser beams. The laser beams excite the fluorescent dyes and result in the emission of specific spectral waves, colored light, which are recorded by a specific photoelectric device. The data thus generated as a fed to a computer where the emission data from the gel is converted into a corresponding nucleotide sequence of the DNA sample. The nucleotide sequence is also represented in specific peaks indicating each nitrogen base. Maxim-Galbert method. In 1976-1977, Alan Maxim and Walter Galbert developed a DNA sequencing method which is also a chemical cleavage method because it is based on chemical modification of DNA and subsequent cleavage at specific bases. Procedure. The DNA to be sequenced must first be end level at one end only. This is accomplished by kinase treatment with 32P ATP which transfers radioactive phosphorus from ATP to 5 end of each strand. Dimethyl sulfoxide DMSO is then added and the labeled DNA samples heated to 90 degrees centigrade. This results in the breakdown of base pairing and dissociation of DNA molecule into its two component strands. The two strands are separated from one another by gel electrophoresis. One strand is purified from the gel and divided into four samples G, A plus G, T plus C, and C, each of which is treated with one of the cleavage regions and modifying chemical regions. The former are used to cut DNA strand at specific point, while the latter cause chemical modification in the nucleotide. They are specific for making them too susceptible to cleavage. DNA analysis. DNA analysis or DNA fingerprinting is an examination method that emerged in the 1980th century and is credited to Alec Jeffrey, an English geneticist who made first DNA fingerprint in 1985. Every species has unique genetic sequences. DNA analysis allows any type of organism to be identified by analyzing its genetic sequences. The method can also be clarify questions of identification within a species. Identification within a species can present more of a challenge than determining between two different species. For example, it is much easier to determine whether a victim was attacked by a bear or human than it is to determine which human perpetrated an attack. Human DNA not only has a record of each person individually, but also a shared history of the evolution of our species and a code that can provide insight into person's future health. So today, DNA analysis has become a standard practice for defining paternity and maternity, predisposition to disease, embryonic health, criminal guilt, or innocence, and so on. DNA can be analyzed in various ways for these purposes. Procedure. There are several techniques that can be used for DNA analysis. Structure fragment length polymorphism RFLP was one of the first methods used in DNA analysis. Following are the key steps to make a DNA fingerprint by using this method. Collection of DNA samples. For DNA analysis, very small fraction of DNA is sufficient because it can be amplified several times with the help of PCR. Therefore, it can be collected even from a small trace of blood or from the cell of single hair root. DNA sample can also be collected from mummified organisms or from fossils when evolutionary relationship has to be studied. Placement of RFLP. RFLP refers to the different size fragments of DNA produced by a particular restriction enzyme. Every person has a unique set of RFLPs because the restriction site of a particular enzyme is always different in number and distribution in all human on Earth except the monozygotic identical twin. Therefore, RFLPs of any two persons, when compared, one can easily analyze their individuality. However, the entire human have 99% similarity in nucleotide sequence of their genome. This is the only 1% difference in genome sequence that establishes the individuality of every person. Placement of RFLP as a digestion of DNA samples by a particular restriction enzyme, which produces a set of different size DNA fragments, RFLPs. Separation of RFLPs. The DNA fragments are then electrophoresed or an agarose gel to separate them by size. The mixture of RFLPs is loaded in polyacrylamide gel and runs for electrophoresis. Fragments of various lengths begin to move at different rate from negative to positive pole within the gel. When the movement is stopped, the gel is proceeded for further treatment in order to observe bending pattern. Southern blotting. Southern blot is a method routinely used in molecular biology for detection of specific DNA sequence and DNA samples. Southern blotting is the combination of two steps: transfer of electrophoresis, separated DNA fragment to a filter membrane, and subsequent fragment detection by probe hybridization. The method is named after its inventor, the British biologist Edwin Southern. Autoradiography. After hybridization, access probes are washed from the membrane and the pattern of hybridization is visualized on X-ray film by exposing the membrane to an X-ray source. This technique is known as autoradiography. The bending pattern which was originally obtained in the gel due to the separation of RFLPs, is now developed on an X-ray film. Application of DNA analysis. Today, DNA analysis has wide range of application and different fields apply. It can be used to identify potential suspects whose DNA may match evidence left at crime scenes. Main important point: exonerate person wrongly accused of crimes, identify crime and catastrophe victims, establish fraternity and other family relationships, identify endangered and protected species as an aid to wildlife officials could be used for prosecuting poachers, detect bacteria and other organisms that may pollute air, water, soil, and food, match organ donors with recipient and transplant programs, determine pedigree for seed of livestock breeds. Genome maps. The genome is a collection of all the genes found in one complete set of chromosomes. So a plant organism has two copies of genome, while egg or sperm has one. Genome maps, just like the road map and street map of a city, which guide us to reach a specific location. The genome maps are used by the scientists searching for a specific gene somewhere within the vast human genome. They have available to them two broad categories of maps: genetic maps and physical maps, which are being used for genome analysis. A genetic map, like an interstate highway map, provides an indirect estimate of distance between two loci. On the other hand, physical map mark estimate of the true distance measurement called base pairs between location, loci, interest, or loci. To continue our analogy, physical maps would then be similar to street maps, where the distance between two sites of interest may be defined more precisely. Genetic markers. Just like interstate maps have cities and towns that serve as landmarks, genetic maps have landmarks known as genetic markers or markers for short. The term marker is used very broadly to describe any observable variation that results from an alteration or mutation at a single genetic locus. A marker may be used as one landmark on map. In most cases, that stretch of DNA is inherited from parent to child according to the standard rule of inheritance. Markers can be within the genes that code for noticeable physical characteristics such as eye color, or a not so noticeable traits such as disease. DNA-based regions can also serve as markers. These types of markers are found within the non-coding regions of genes and are used to detect unique regions on a chromosome. DNA markers are especially useful for generating genetic maps when there are occasional predictable mutations that occur during meiosis, the formation of gametes such as egg and sperm that, over many generations, lead to high degree of variability in the DNA content of the marker from individual to individual. Commonly used DNA markers: RFLPs are restriction fragment length polymorphism. VNTRS are variable number of tandem repeat polymorphism. Microsatellite polymorphism. SNPs are single nucleotide polymorphism. Genome analysis. Due to rapid development of genome studies, a new branch of biotechnology has emerged called genomics, which deals with exploration and analysis of complete DNA sequence of an organism genome. This field was not really possible until the publication of the genome of Haemophilus influenzae as in 1995. Before this, Pete Singer also had sequenced the first bacteriophage genome for which he later won the Nobel Prize. Besides, a Human Genome Project was also launched in 1990. Human Genome Project (HGP). The Human Genome Project is an international scientific research project which is based on the exploration and analysis of human genome. It was originally founded by the US Department of Energy and the National Institute of Health in 1990. They have established National Human Genome Research Institute (NHGRI), who has completed this task in 2003. James D. Watson was appointed as first director of this institute, but at the time of completion of the project, the institute was being led by Dr. Francis Collins. Although this project was funded initially by the US government, later on Wellcome Trust UK became a major partner. Additional contribution came from Japan, France, Germany, China, and others. Major goals of HGP. Major goals and objective of this project were to identify all the approximately 20,000 to 25,000 genes in human DNA, determine the sequences of the 3 billion chemical base pairs that make up human DNA, store this information in databases, improve tools for data analysis, transfer related technologies to the private sector, and address the ethical, legal, and social issues that may arise from the project. Benefits of HGP. Some potential benefits from Human Genome Project are expected in the following fields: molecular medicine, improved diagnosis of diseases, earlier detection of genetic predisposition to the disease, rational drug design, gene therapy and control system for drugs, pharmacogenomics, custom drugs, bioarchaeology, anthropology, evolution, and human migration. Study evolution through germline mutation and lineages. Study migration of different population groups based on female genetic inheritance. Study mutation on Y chromosome to trace lineage and migration of males. Compare breakpoints in the evolution of mutations with ages of populations and historical events. Tissue culture, most important term in biology and biotechnology. The propagation of a plant by using a plant part or single cell or group of cell on a test tube under very controlled and hygienic condition is called tissue culture. Tissue culture is often a generic term that refers to both organ culture and cell culture. The initial plant part which is used to develop tissue culture is called explant. It may be complete organ, seed, leaf, and twig, a single cell protoplast, or a piece of tissue on the basis of explant. Tissue culture is variously called as cell culture or organ culture. Procedure of tissue culture. A typical tissue culture method consists of the following steps: sterilization. Tissue culture is performed under aseptic condition. Sterilization refers to the decontamination because living plant material from the environment are naturally contaminated on their surfaces and sometimes interiors with the microorganisms. So, surface sterilization of starting material, explants, and chemical solution, usually sodium or calcium hypochlorite or mercury chloride, is required. The glassware which is to be used in the procedure should also be sterilized. A sterilization machine. Media preparation. Cell is natural medium of plant growth which is supposed to be very complex for tiny plant. So, in tissue culture technique, plants are grown on artificial media in which composition is kept under control. Solid or liquid media are used depending upon the need, which are generally composed of inorganic salts, plus a few organic nutrients, vitamins, and plant hormones. Inoculation. Inoculation refers to the placement of explant to the surface of solid culture medium, but in sometimes placed directly into a liquid medium, particularly when seal suspension cultures are desired. This is done in laminar flow, a sterile chamber. After the placement of explant, it is ensured that petri plates or test tubes should be airtight. Next, these glass wells are shifted to the growth room or incubator. Development of callus. First, the plant is allowed to grow into the unorganized mass of cells callus, which is then shifted to the new media for the development of shoots and roots. A balance of both auxin and cytokinin will often produce an unorganized mass of cells, the callus. Development of plantlets. As the callus grows, pieces are typically sliced up and transferred to new media subculture to allow for growth or to alter the morphology of the callus. The skill and experience of the tissue culturists are important in judging which species to culture and which to discard. As shoot emerge from a culture, they may be sliced up and rooted with auxin to produce plantlets, which, when mature, can be transferred to potting soil for further growth in the greenhouse as normal plants. Types of tissue culture. There are several types of tissue cultures which are primarily based upon type of explant used. Callus culture. When explants are cultured in the appropriate medium, usually with both enoxine and cytokinin, can give rise to an unorganized growing and dividing mass of cells. This is called callus culture. Any plant tissue can be used as an explant. Seal suspension culture. Seal suspension culture is developed from the callus when it is placed into a liquid medium and then agitated single cell and a small clumps of cells are released into the medium. Under these cells continue to grow and divide, eventually producing a cell suspension culture. It can be scaled up by repeated subculturing into fresh medium. Large cell clumps can be removed during subculture of the cell suspension. Cell suspension cultures are very useful to obtain some drug compounds which are generally obtained from specific part of adult plant because the cell suspension culture produce the same chemical as the entire plant. For example, cell suspension culture of Cinchona legriana produce quinine and those of Digitalis lanata produce digitoxin. Protoplast culture. Protoplasts are plant cells with the cell wall removed. Protoplasts are most commonly isolated from other leaf mesopel cells or cell suspension. Protoplast cultures can be used to develop whole plant by organogenesis or somatic embryogenesis, synthetic embryos that are developed from somatic cells. Genetic variation can also be induced in these somatic embryos if they are exposed to chemical or physical mutagens. Such variations are called somaclonal variations. Protoplasts also act as ideal targets of transformation by a variety of means: callus, cell suspension, cell transformation. Meristem culture. Meristem are the rapidly dividing and growing tissues, especially found at the apices of roots or shoots and in some other plant parts. A tissue culture in which meristem are used as explant is called meristem culture. Generally, root apical or shoot apical meristem are used to meristem culture. Meristem culture is mostly used for macropropagation and to obtain virus are parasite-free plant because the whole plant may be infected by virus, bacteria, or fungi, but meristems are devoid of viruses due to the absence of vascular system in them. Interculture. Interculture is also called as microspore culture or pollen culture. It is a technique in which mature anthers or microspores are cultured in a suitable medium. The haploid cells tube cells present in the microspore begin to divide and produce a plant callus. A plant callus can be grown into a plant plant, and a plant plant can also be obtained a chromosomal doubling as induced by the colchicine treatment. Animal cell culture. An important aspect of any biotechnological processes is the culture of animal cell and artificial media. These animal cell and culture are used in recombinant DNA technology, genetic manipulations, and a variety of industrial processes. Nowadays, it has become possible to use the cell and tissue culture in the areas of research which has a potential for economic value and commercialization. The animal cell cultures are being extensively used in production of vaccines, monoclonal antibodies, pharmaceutical drugs, cancer research, genetic manipulation, etc. Techniques of animal cell culture. Animal cell can grow in simple glass or plastic containers and nutritive media, but they grow only to limited generation. A cell culture, which is initiated by the cell removed from an animal organ, is called primary cell culture, whereas the primary culture is subcultured in fresh media to establish secondary cultures. The culture of native tissue that retains most of in view astrological feature is regarded as organ culture, while the culturing of the cell of their reaggregation to form a tissue-like structure represent histotypic culture. Another culture technique involves the recombination of different cell types to form a more defined tissue or an organ known as organotypic culture. Among the essential requirements for animal cell culture are special incubator to maintain the level of oxygen, carbon dioxide, temperature, humidity as present in the animal body, and the synthetic media with vitamins, amino acid, and fetal calf serum. Synthetic media are prepared artificially by adding several organic and inorganic nutrients, vitamins, salts, serum, protein, carbohydrates, cofactors, etc. Different types of synthetic media can be prepared for a variety of cells and tissue to be cultured. Synthetic media are of two types: serum containing media, media containing serum, and serum free media, media without serum. Application of animal cell culture. The animal cell culture are used for a diverse range of research and development. These areas are production of antiviral vaccines, genetic manipulation, which is easy to carry out in cell or organ cultures, production of monoclonal antibodies, requires cell lines in culture, production of pharmaceutical drugs using cell lines, chromosome analysis of cells derived from womb, study of the effects of toxins and pollutants using cell lines, use of artificial skin, study the function of the nerve cells. Transgenic organisms. Combining genes from different organisms is known as recombinant DNA technology, and the resulting organism is said to be genetically modified, genetically engineered, or transgenic. In other words, the free living organism in the environment that have had a foreign gene inserted into them are called transgenic organisms. Bacteria were the first transgenic organism. First transgenic bacterium was produced in 1973. Since then, many transgenic organisms, such as animals, plants, and bacteria, have been produced. Genetic modification consists of a special set of techniques that alter the genetic makeup of the organisms. Transgenic bacteria. Bacteria were the first organism to be modified in laboratory due to their simple genetics. The first example of this occurred in 1978 when Herbert Bayer, working at the University of California laboratory, took a version of the human insulin gene and inserted into the bacterium Escherichia coli to produce synthetic human insulin. Rule of transgenic bacteria in making biotechnology products. Transgenic bacteria are now being used in a variety of ways and are particularly important in producing large amounts of pure human protein for use in medicines. Genetically modified bacteria are used to produce the protein insulin to treat diabetes. Similar bacteria have been used to produce clotting factor to treat hemophilia and human growth hormone to treat various forms of dwarfism. Some transgenic bacteria have been produced that synthesize tissue plasmo, plasminogen activator (TPA), a protein used by heart patients to treat thrombotic disorder is a dissolved clotted blood masses and anterior interferons which are used for treating viral infection. These recombinant proteins are much safer than the product that replaced since the older products were purified from claders and could transmit diseases. Transgenic bacteria are also being used for bioremediation, removal of environmental pollutant from organism by organism. Such bacteria are also used to clean up and recovery from an oil spill. Ecological concerns surrounding transgenic bacteria, where lots of benefits are being obtained from genetically engineered bacteria. There are some ecological concerns are associated with these bacteria. One of the main issues regarding this is the possibility that hazardous new pathogens might be created. Transgenic plants. The first field trials of genetically engineered plants occurred in France and USA in 1996 when tobacco plants were engineered to be resistant to herbicides. In most cases, the aim of developing transgenic plant is to introduce a new trait to plant which does not occur naturally in this species. Examples include resistance to certain pests, diseases, or environmental conditions, or the production of certain nutrients or pharmaceutical agents. Methods of gene transformation in plants. There are many techniques of gene transformation have been developed. In all techniques, gene of interest is introduced into single or few plant cells, which are then allowed to regenerate in a suitable culture medium. Upon a successful culture procedure, a transgenic plant is obtained. Agriculturally improved transgenic crops. During the last 2 decades, a tremendous progress has been made in the development of transgenic plants using the various techniques of genetic engineering. As per estimate recorded in 2002, transgenic crops are cultivated worldwide on about 148 million acres, 587 million hectares land by about 5.5 million farmers. Transgenic plants have many beneficial traits like tolerance against biotic stresses, viral, bacterial infection, pests, and weeds, and biotic stresses, physical actors such as temperature, humidity, salinity, drought, waterlogging, etc. We decide our herbicide tolerance, for example, glyphosate resistance and paspinothisin resistance. Insect resistance like transgenic crops with Bt genes, cotton, rice, maize, potato, tomato, brinjal, cauliflower, cabbage, etc. Delayed fruit ripening, improvement in nutritional contents. Some of the commercially grown transgenic plants in developed countries are Roundup Ready, soybean, Freedom 2 squash, high lauric, Refreshed canola, Flavor sour, and endless summer tomatoes. During 1995, full registration was granted to genetically engineered wheat gene containing insect-resistant new leaf potato, maximizer, corn, Bollgard cotton in USA. In July 2000, researchers from the team that produced Jolie reported success in producing transgenic lambs in which the transgene had been inserted at a specific site in the genome and functioned well. Transgenic animals. Transgenic animal is one that carries a foreign gene that has been deliberately inserted into its genome. Transgenic animals have the potential to improve human welfare in agriculture, such as larger sheep that grow more wool, medicine, such as cows that produce insulin in their milk, industry, such as goats that produce spider silk for materials production. Methods of creation of transgenic animals. In comparison to that, for larger vertebrates, mice have become the model animal used in field of transgenics because of their small size and low cost of housing, short generation time, and fairly well-defined genetics. The insertion of gene is high over random process, and there is high probability that introduced gene will not insert itself into a site on the host DNA that will permit its expression. The three principal methods used for creation of transgenic animals are DNA, microinjection, embryonic stem cells mediated gene transfer, and retrovirus mediated gene transfer. Biotechnology and healthcare. The tools and techniques of biotechnology have opened up new doors when it comes to researching and learning more about the human body and what goes wrong, health and disease. This has led scientists to improve methods of treating and preventing those diseases. Biotechnology has made a huge difference in human healthcare and has now enabled scientists to develop products which can give quicker and more accurate tests, therapies, and that have a lot less side effects and vaccines which are safer than ever before. Development of vaccine and biotechnology. Biotechnology is used in three different ways in the development of vaccine. Separation of pure antigen using a specific monoclonal antibody. Synthesis of an antigen with the help of a cloned gene. Synthesis of peptide to be used as vaccines. Rule of biotechnology and diagnosis of diseases. Many human diseases can be diagnosed by using products of biotechnology like monoclonal antibodies and DNA RNA probes. Monoclonal antibodies. The response of the immune system to any antigen, even the simplest, is polyclonal. That as the system manufactures antibodies of a great range of structure both in their binding region as well as in their effector region. Monoclonal antibodies MAB are a group of identical antibodies because they are made by identical immune cells that are all clones of a unique parent cell. Given almost any substance, it is possible to produce monoclonal antibodies that specifically bind to that substance that can then serve to detect and purify that substance. This has become an important tool in biochemistry, molecular biology, and medicine. Not only can antibodies be used therapeutically to protect against diseases, they can also help to diagnose a wide variety of illness and can detect the presence of drugs, viral and bacterial products, and other unusual or abnormal substances in the blood. Monoclonal antibodies are typically made by fusing myeloma cells, cancerous B lymphocytes, with the spleen cells from mouse that has been immunized with the desired antigen. The technique is called somatic cell hybridization. DNA RNA probes. In biotechnology, a probe is fluorescent or radioactive labeled fragment of DNA or RNA of variable length, usually 100 to 1000 bases long, which is used in DNA or RNA samples to detect the presence of nucleotide sequences that are complementary to the sequence in the probe. Such probes are widely used in diagnosis of many viral and bacterial diseases. Diagnosis of diseases caused by protozoa and helminthes. The monoclonal antibodies and DNA probes are being used as very sensitive tools in biotechnology to diagnose the diseases caused by protozoa and helminthes. Monoclonal antibodies can be used through serological tests which takes only minutes as compared to conventional method which require some weeks as well. as the bacteria and viruses have to be cultured. For example, in herpes virus, the DNA probes are more sensitive than monoclonal antibodies, and the process takes hours instead of weeks. Ready-made DNA probes for herpes virus and other human, animals, and plant viruses are being prepared. Probes are now available for a number of human parasites from the group Protozoa and Helminthes. Gene therapy. Gene therapy is a technique for correcting defective genes responsible for disease development. Researchers may use one of several approaches for correcting faulty genes. A normal gene may be inserted into non-specific location within the genome to replace a non-functional gene. This approach is most common. An abnormal gene could be swapped for a normal gene through homologous recombination. The abnormal gene could be repaired through selective reverse mutation, which return the gene to its normal function. The regulation, the degree of which a gene is turned on or off a particular gene could be altered. Mechanism of gene therapy. In gene therapy treatment, normal gene is either delivered directly into the body and view or into the cell outside the body. Then these transgenic cells are again implanted into the body ex vivo. In both cases, the normal gene is inserted into the genome to replace abnormal disease causing gene. A carrier molecule called vector must be used to deliver the therapeutic gene to the patient target cells. Currently, the most common vector is a virus that has been genetically altered to carry normal human DNA. Viruses have evolved a way of encapsulating and delivering their genes to human cells in a pathogenic manner. Scientists have tried to take advantage of this capability and manipulate the virus genome to remove disease causing genes and insert the therapeutic genes. Target cells such as the patient liver or lung cells are infected with the viral vector. The vector then unloads its genetic material containing therapeutic human gene into the target cell. The generation of functional protein product from the therapeutic gene restores the target cell to a normal state. Beside the virus-mediated gene delivery system, there are several non-viral options for gene delivery. The simplest method is the direct introduction of therapeutic DNA into target cells. This approach is limited in its application because it can be used only with certain tissues and requires large amount of DNA. Another non-viral approach involves the creation of artificial lipid sphere within aqueous core. This liposome, which carries therapeutic DNA, is capable of passing DNA through the target cell membrane. Cystic fibrosis. An inherited disease, cystic fibrosis affects the mucous and sweat gland. People with severe symptoms can have serious lung and digestive problems, while people with a mild form of the disease may not have any symptoms until they are adolescents or young adults. Normally, mucus is watery. It keeps the lining of certain organs moist and prevents them from drying out getting infected. However, the cause of cystic fibrosis is a defect in the cystic fibrosis transmembrane conductance regulator CFTR gene that encodes a protein by which the movement of salt and water is controlled in and out of your cells. In people with cystic fibrosis, the gene does not work effectively. As a result, cells that line the passageways of the lungs, pancreas, and other organs produce abnormally thick, sticky mucus. This mucus obstructs the airways and glands, which causes the characteristic signs and symptoms of cystic fibrosis. Gene therapy of cystic fibrosis. In 1999, experts discovered the gene that causes cystic fibrosis, identified it as the cystic fibrosis transmembrane conductance regulator or CFTR. The discovery of this defective gene forced New possibilities occur. Experts proposed gene therapy as plausible method for curing the disease. Gene therapy is the process of creating a healthy version of the flawed CFTR gene and infusing it into the affected cell in the body, particularly into the lungs of cystic fibrosis infected patients.